the Great Barrier Reef. Every day, thousands of tourists are thrilled by its wild beauty.
I love it. It's kind of scarier than I thought it would be.
I didn't think it was going to be so beautiful, even just the coral. It's been on the bucket list for a while. Just thought it was about time before it gets any more degraded.
Large parts of the barrier reef are suffering the worst coral bleaching Australia has ever seen.
Personally, I've been devastated. I've come out of dives and broken down in tears from what I've seen.
Our reef, like reefs worldwide, is struggling to survive global warming. A change of just a few degrees spells the difference between life and death.
Coral reefs have been thriving on this planet for hundreds of millions of years. But if climate trends continue, this century could be their last.
As scientists race against time, we explore why bleaching is a mortal threat to the Great Barrier Reef and the challenges we all face to protect this global treasure. Animal, plant, or mineral? Coral is a bizarre mix of all three. An upside-down jellyfish called a polyp that embeds plant cells in its flesh and builds solar power cities from limestone. It seems so alien that Brett Lewis muses he wouldn't be surprised if life on another planet looked like this.
A beautiful coral planet? Why not? Just a whole thing, just a big calcium carbonate planet.
His time-lapse videos reveal the secret life of corals.
They're not static, and that's absolutely what our videos are able to show, and what our research shows is just how dynamic they actually are.
To survive, coral needs a key partner, algae. Microscopic single-celled algae of the genus Symbiodinium. Coral takes the algae from the water to live symbiotically inside its own cells. That's how the polyp gets its colour. It's a great relationship. The algae, or symbionts, receive shelter and carbon dioxide from their host. In return, the coral gets most of its nutrition from sugars that the algae make through photosynthesis. But there's a catch. This solar-powered partnership depends on temperature to work. So what happens when water warms up? Over a week, QUT researcher Brett Lewis increased the water temperature by 4 degrees to peak at 32 degrees Celsius.
That's what we wanted to test, see what the response of corals were at that upper limit and see how they're going to cope.
After 24 hours, this is what he saw.
It's really violent. It's much more violent than we expected.
Mushroom corals, a large solitary type that don't build reefs, expelled their algal symbionts with repeated convulsions known as pulsed inflation.
Some of the largest expansions we've seen were 340% the size of the actual original tissue.
As clouds of algae are pumped into the water, the coral loses its colour and becomes pale. Corals are known for doing this to get rid of sand, but getting rid of algae in this way has not been seen before.
This is the first time we've ever been able to document pulsed inflation as a way to remove Symbiodinium from the tissue.
That's the reaction of just one coral in a lab. This is what happens on the scale of a reef. Early in 2016, high sea temperatures over many weeks caused mass bleaching in parts of the Great Barrier Reef. Professor Terry Hughes led a massive effort to map what happened.
We flew over nearly 1,200 reefs between the Torres Straits and the southern tip of the Great Barrier Reef. It took 11 flights, about 8 hours of flying a day. It was quite grueling and exhausting and emotional, because we saw hundreds of reefs that were incredibly badly damaged.
Worst hit were the most remote and pristine reefs in the north along Cape York Peninsula.
It's very confronting to see a beautiful coral reef with 80% plus of its corals bleach, because when that level of bleaching occurs, you're looking at 50% or more mortality.
Bleaching doesn't usually kill them outright, but if the algae aren't replaced, the corals slowly starve. Coral biologist Tracy Ainsworth examines corals from the northern reefs with a laser microscope that reveals their details in fluorescent 3D.
What we wanted to do is look at what normal healthy tissue looks like, and then to see just how damaged the coral tissues have been from the bleaching event.
The algal symbionts show as red, the coral tissues as blue and green. When healthy, each square centimetre of coral tissue is packed with around 2 million algal cells.
So if we look here at normal healthy tissue and then compare that to the bleached tissue, all the symbionts are gone. You're not seeing any of the red cells within the tissue. The tissue is really thin and there's not so much fluorescence actually in the coral tissue that's left.
In previous bleaching events, they've seen algal symbionts reduced to about 200,000, a tenfold decrease. This time in samples from the northern reefs, they found barely any left at all.
They're the lowest densities ever recorded. They're lower even than laboratory-based bleaching experiments where people really torture corals. You know, they put them into +4 degrees centigrade. They're the sickest corals we've ever seen.
When bleaching happens, what's going on inside the coral?
The algae actually go into hyperdrive to some extent. So with all that heat, with all that stress, they become overreactive, and therefore the coral doesn't like that. So they essentially just eject them out of their tissues.
Above 30 degrees, the algae start to lose their ability to convert solar energy. That energy has to go somewhere and ends up creating reactive forms of oxygen, like peroxide in bleach, inside the coral cells. The very light a coral needs for growth becomes poisonous.
Bleaching is immediate stress, and we see a lot of mortality, which is really quite tragic for the reef. But there's also all these knock-on effects. So 6 months, 12 months down the track, we're probably going to see more disease.
Once the health of the coral is compromised, bacteria and other microbes cause infections like this.
This lesion will move, up to 2 to 5 millimetres a day. In some areas, it can be seen to move centimetres a day. And so some corals that have been infected with these lesions will be dead within weeks.
For corals to recover, they don't just have to take up their symbionts again. They have to repair their tissues, they have to fight off these invaders, and then they have to select that one microbe that they need to survive.
In 2016, however, many corals didn't get that chance.
What we saw at the peak of the bleaching was a big pulse of more or less instantaneous mortality due simply to the heat stress. So those corals didn't die slowly of starvation because they'd lost their symbionts. They actually cooked over a period of just a week or two because the temperatures in the northern barrier reef were so extreme.
The Bureau of Meteorology has records of ocean temperature stretching back to 1900. The areas of dark orange on this map around the northern Great Barrier Reef and Tasmania are pools of hot water. Here, the average sea surface temperatures this summer were the highest ever recorded.
This year we saw some locations well over 2 degrees Celsius warmer than they would usually experience in the hottest time of year.
Scott Heron works with the US National Oceanic and Atmospheric Administration. This colour-coded map shows the heat stress that bleached the Great Barrier Reef this summer.
Orange is now we're actually starting to accumulate stress.
Red, alert level 1.
We expect bleaching of the more susceptible species, and the darkest red, we expect some of those susceptibles to begin to die, and even the hardiest of species beginning to bleach.
So March was the toughest month.
March was the toughest month.
Although the Great Barrier Reef was hit this year, a global bleaching event has rolled across 3 oceans since 2014.
It wasn't just the El Nino. 2014 was the warmest year on record, an over 100-year record. 2015 exceeded that record by a new margin, and we have estimates of a 99% chance that we're going to break that record again. If you are 30 years or younger, you have never experienced a normal temperature month.
The worry we have is that these events are now becoming so frequent that the gap between them, the return time, is getting shorter and shorter. The median return time now throughout the Indo-Pacific is only 6 years. We know that it takes 10 to 15 years for the fastest growing corals to bounce back after a severe disturbance like a bleaching event or a cyclone. So 6 years is certainly not enough for those corals to recover.
Nine months after the bleaching began, it's time to start assessing the death toll. 100 kilometres north of Cooktown, off Lizard Island, this research vessel is heading out to identify where the surviving corals are. Over the last 30 years, the Australian Institute of Marine Science has been monitoring the condition of the Great Barrier Reef. Teams of divers have been surveying coral cover along the entire 2,000-kilometre length of the reef for decades.
We, as the bleaching response, we target sites that have been studied long-term, and that gives us the ability to detect the change and the mortality and the true impacts from this 2016 bleaching event.
In teams of 4, divers roll out a 50-metre tape and swim 5 transects at each site. One counts large fish, another takes photos at regular intervals, another notes the amount of coral cover and any juvenile coral starting to regrow. As the tape is wound up, another counts small fish. Then comes the manta tow, where an observer is towed behind a small boat at 6 kilometres an hour. Since 1985, Ames divers like Bluey Miller have clocked up more than 30,000 kilometres over 500 different reefs.
I think I might have the Guinness Book of Records. I've done so many I couldn't even count them, but it'd be tens of thousands.
Tens of thousands of manta tows?
Yeah, each manta tow goes for 2 minutes, and then as we go along the edge of the reef getting towed behind the boat, after 2 minutes we stop and write down our observations.
To avoid disputes over data, it's important that survey techniques are standardised. Sections of the Queensland tourism industry, worried that bad news may deter tourists from visiting the reef, have claimed the bleaching results are a great white lie.
The report will just simply continue to give oxygen to the theory that the Great Barrier Reef is dead and dying. It is just simply not true.
If you just randomly go to a reef location 6 months later, you're going to see survivors, and we hope to see surviving corals from this event. But you need to have a meticulous method of assessing bleaching in a site and then returning to the site to say whether there is severe impacts of a bleaching event or not.
The big question is there enough time for these reefs to recover between these impacts?
Are you optimistic?
I'm optimistic. I'm a glass half full guy.
But with what he saw from the aerial surveys in the north, Terry Hughes knows recovery could take decades.
800 kilometres of the Barrier Reef in the northernmost section from New Guinea down to Port Douglas has lost half of its coral. It's as if 10 Category 5 cyclones holding hands have walked ashore north of Cairns.
Ironically, it was one of those cyclones that prevented the destruction extending the entire length of the Barrier Reef. Severe tropical cyclone Winston was the strongest to hit the South Pacific in recorded history. Tragically, when it struck Fiji in February, 44 people were killed. 40,000 houses were damaged or destroyed.
It came eventually to the coast of Queensland as a rain depression, and it sat around the bottom half of the Great Barrier Reef for a period of several weeks. It brought the temperature down by about 2 degrees centigrade. So the Barrier Reef was saved, the southern half, by the vagaries of that cyclone coming along.
Which brings me here to the southern Great Barrier Reef off Gladstone. Heron Island and its research station holds a special place in my heart.
The last time I visited Heron Island was as a high school student way back in 1980. The beauty of that first reef dive changed my life forever.
I'll never forget that feeling of awestruck reverence. Thankfully, the reef here has been spared the damage further north.
A relief after this year's bleaching event?
It is certainly a relief. It's fantastic to see such a vibrant ecosystem.
And it's only because of that sort of last-minute reprieve that we're able to see it still in its glory now.
Look, and weather events like that happen to be able to mitigate what could have been, and it's a delight to see such a thriving place.
Yeah, too right.
Physical scientist Scott Heron and biochemist Bill Leggett are part of a team experimenting with the abilities of coral to recover from bleaching. It means taking healthy corals from the reef flat and exposing them to the same conditions as their northern counterparts had to endure.
When it comes to climate change, the big question is this: how much heat stress can corals tolerate, for how long and how often, before they reach a tipping point from which they can no longer recover?
Right, so this is one of the acropers that we'll be working on, and we'll just give it a gentle tap, and then we can get a small piece of him out.
Just a love tap.
Just a little love tap. And then we can take him back to the island and he can be one of our experimental subjects.
How many more samples do you reckon you're going to need?
Oh, I think we might have enough now.
So today's the day you start?
We will. So we'll start getting these guys settled in, and then we'll start putting the temperature up, and we'll see how they go.
The experiment will run for 8 weeks to simulate the duration of this year's bleaching event.
Temperature tanks are going to mimic what happened on the northern GBR this year, and the two controls allow us to compare what the responses are of corals normally.
They're also testing how temperature and sunlight interact to cause bleaching.
We've got a series of tanks here that are going to be exposed to light, and then behind me we've got a series of tanks that are going to be in the shade. And then after about 3 or 4 weeks we're going to switch them over, and we're going to put all of these through a simulated bleaching event again.
How hot are you making the water in this experiment?
So the average for Heron Island's about 28 degrees, so we'll take it up to about 32 degrees for this experiment.
Temperature records from the Great Barrier Reef over the last 27 years show that most of the times water got hot enough to bleach coral, the way it warmed actually made them more heat resistant.
And that small pulse of temperature, a little bit of early warmth and then a cool off, was preconditioning the corals to respond better when the big stress came.
But when they model future warming, those protective pulses disappear with an increase of just ½ degree, or within 4 decades at current rates. Without them, bleaching events will become much more lethal for corals.
As temperature goes up, the first peak goes up, then the relaxation period goes up too. And so instead of it being a relaxation period, that recovery section actually moves into the stress zone, the stress responses.
So it never gets to relax?
So it never gets to relax.
With the climate change and the CO2 we've already emitted, we're committed to seeing increasing temperatures for the next 30 to 40 years. I don't think corals are going to be able to keep up with that.
The future of the entire Great Barrier Reef and reefs worldwide rests on the balance between life and death inside the cells of coral.
This little guy here will get express shipped down to UTS, our collaborators, and be able to analyse what he's doing in the lab.
In downtown Sydney, marine biologists like Dave Suggett want to discover the microbes that work best for coral and why.
We now know that Symbiodinium has a broad range of genetic diversity, in fact, as many as 400 or 500 species. In fact, the deeper we go into the genetic variation, the more variability we find.
Unexplored territory, really.
Completely unexplored territory. We're really on the front line with this.
The question is, will this genetic diversity be able to match the challenges of environmental change?
We're literally scratching the surface in terms of Symbiodinium. In this room, we probably have about 20 species.
Like a breathalyzer for corals, this is a mass spectrometer that measures the gases they breathe in and out. Samples from Heron Island are tested here to see how the performance of their symbiotic algae is affected by the bleaching experiments.
As they become less efficient, they start drawing less oxygen, carbon dioxide levels start to go up, and we can monitor all of that in absolute real time and monitor or record the collapse of the symbiosis.
To what extent can corals save themselves by selecting new symbionts, new solar panels, if the old ones aren't up to scratch?
When the corals reproduce and they create baby corals or planular larvae, they settle onto the substrate and they're open to take up all this diversity of symbionts in nature, and they do that actually. We now know that they'll acquire a large number of species initially, and then over time it seems to select for a species of symbiont that it's learned to evolve to live and operate with most successfully.
Incredibly, there's emerging evidence that if they survive a bleaching event, some adult corals can then switch their algal symbionts to a tougher species.
We can find that little trick, that little trait that these symbionts have acquired to make them either sensitive or tolerant. We can start searching for that more broadly in the ecosystems.
Like in the Red Sea and Persian Gulf, where corals live in high salinities and temperatures up to 36 degrees, well beyond what corals on the Great Barrier Reef can tolerate. Genetic sequencing of these species and their algae may reveal clues to how they survive.
So, in a way, the secret to the future of corals could be here with you right now.
Absolutely, absolutely. We're still starting to learn just what secrets these algae have and really what they'll bring to the future of reefs.
As reefs degrade, scientists are asking a tough question. Should we stand back and let nature take its course, or step in and give evolution a helping hand?
This is the National Sea Simulator near Townsville, the largest, smartest aquarium in Australia. Now, if we're running a global experiment on changing the climate, it makes sense that we need an industrial scale facility like this to see what those changes mean for coral.
Here, corals can be exposed to the hot, acidic ocean conditions that are projected for later this century. Madeline Van Oppen's team is working on a big idea.
The main aim is to enhance the tolerance of corals to environmental stress, and mostly stress related to climate change.
They want to create supercorals with super symbionts that can tolerate higher temperatures without bleaching. This assisted evolution, as they call it, is Leela Chakravarti's PhD project.
We are taking these algae and we're exposing them to higher temperatures over a number of generations. So it's the same as natural selection, but we're doing it at a much faster pace so that we can get them to evolve in short time periods.
It takes only 150 quick generations for the algae to evolve the ability to grow faster and bleach less when the temperature goes up by 4 degrees. The next step is to get them into the coral.
So here is a coral recruit, glowing green, and these little red dots are actually the Symbiodinium that we've added to the water. And after a few days, you can actually start to see that the coral has taken up these Symbiodinium cells.
What, it just absorbs them?
So what it does is it eats them, and it recognises them as being Symbiodinium, so they aren't digested, and then they're taken into the coral tissue where they live.
How cool is that?
Very cool.
The bleaching of coral with the new evolved algae is compared with ordinary coral at 2 different temperatures, 27 degrees and 31 degrees.
We did this for 3 different coral species. We found that the effect of the temperature evolution is a bit smaller, but there was still a significant delay in the bleaching of the corals that associated with the evolved algae compared to the unevolved algae.
It's an exciting proof of concept showing it may be possible to rapidly evolve heat-resistant corals.
I think people describe the science as controversial because they sometimes say you're playing God, because we're manipulating the constitution of the organisms, if you like. But we're not actually using any mechanisms that do not occur naturally. We're just trying to speed up those natural processes of evolution.
But if supercorals can be created, what to do with them? While small areas could be restored, Terry Hughes cautions it's a problem of scale.
No one has ever successfully restored a reef, even at the scale of a kilometre, never mind 800 kilometres. So prevention is better than cure. The bleaching in the northern part of the Great Barrier Reef this time around is so severe that we're losing the toughest corals, the 50-year-old, the 100-year-old corals, and it takes that length of time for those corals to be replaced. So the northern Great Barrier Reef has changed forever.
Out at sea near Cairns, though, the tourists are having the time of their lives.
It's one of the things that y'all brag about, you know, that oh man, you gotta make sure you go to the reef, do some diving and all that other stuff, you know, besides going to see some kangaroos.
Sometimes I think we forget that the reef contributes more than $6 billion and 70,000 jobs to the Australian economy each year.
More than 3/4 of Australians regard the reef as part of our national identity, although many of us have never visited it. It suggests that protecting the reef and keeping it healthy is part of what it means to be Australian.
However, we're living a paradox. We're home to the largest living marine structure on the planet, alongside some of the biggest coal deposits in the world. We're part of an international agreement to limit global warming to under 2 degrees, yet we're also the world's leading exporter of carbon emissions.
2 degrees is enough to save corals. We'll always have corals, but the problem is I don't think 2 degrees is enough to save coral reefs.
It seems inevitable that global warming will eventually force us to choose between coal or coral. And it leaves me wondering, if mass bleaching and reef death doesn't galvanise action on climate change, then what will?
The choice is whether we want to just sit back and enjoy these ecosystems while they're hanging on by a thread, or whether we want to literally dive right in and do something proactive.